Thank you for joining us for today's webinar. This is part 2, The Developing Brain, a neuroscience perspective presented by Dr. Laurie Slifka, Faculty Department of Psychology from the Kaiser University Graduate School.
Hi, everyone. Thank you so much, Dr. Miller, for that introduction. As Dr. Miller said, my name is Dr. Laurie Slifka. I hold a PhD in psychology with an emphasis in behavioral neuroscience from the University of Missouri, St. Louis. Go Blues. And I currently live in South Florida and serve as a full-time faculty member in the psychology graduate department. So this is part 2 of the developing brain. On Tuesday, we discussed how the brain develops from the prenatal stage into the adult stage. We talked about the influence of genes and environment on brain development, as well as some risk and protective factors in terms of brain development. And then the goal of Tuesday was to hopefully start thinking about strategies to help children that have experienced toxic stress and childhood adversity. So today is an extension of that. We're going to go into a little bit greater depth now that we have a foundational understanding of the brain. And we're going to start off by going into like a brief review about what we talked about on Tuesday about the developing brain. So at the end, you should be able to describe how the brain develops in typically developing children and children with neurodevelopmental and some other disorders, and to understand the biological basis of cognitive and emotional function in children and adolescents. And then hopefully this knowledge will help you to develop effective approaches to educate students with and without neurodevelopmental and other disorders. All right, so as I said, I want to start off by providing a brief review. Thank you for those that attended on Tuesday and are back, welcome. For those that weren't able to make it on Tuesday, that is okay. We will go over what we discussed. So just to start off with an introduction of brain development, this is an image of what the brain typically looks like from 3 weeks prenatal stage to birth. So typically when we think of the brain, we think of the lower right-hand picture, but the brain doesn't start off looking like that. This development really starts around 16 days. That's where the foundation of the brain and the spinal cord starts. So that foundation is set in place, and then there's a cascade of events that eventually will change to the brain that we are used to seeing, like in the bottom right-hand corner. Around week 6 or 7 is when it really starts looking like more like a brain, and it's divided into sections. That's the picture on the right top corner. So it's divided into 3 sections called the forebrain, midbrain, and hindbrain. And under that is the development of the spinal cord. Around week 11, there's more rapid development of structures. You can see in the bottom left picture, the enlargement of all of the regions. It's starting to look a little bit more like what we think of when we think of brain. And by the end of the second trimester, which is around 26 weeks, the brainstem is almost entirely mature. The brainstem, which we went over on Tuesday, is an extremely important structure for keeping us alive. It's important for essential functions that are automatic. So things that we're not thinking about that are taking place, like heart rate, breathing, and blood pressure. And that rests just above the spinal cord, but below the cerebral cortex. The cerebral cortex is the big area we think of when we think of the brain, and that's the last area to mature. That's all of our thinking. The brain roughly triples in weight during the last 13 weeks gestation. It grows from about 3.5 ounces at the end of the second trimester to about 10.6 ounces at term. And obviously it starts looking a little bit different too. We can see by this picture right here, the picture in the bottom right corner. So it went from a smooth surface to increasingly grooved and indented. So there's a lot of growth that happens prenatally and growth that actually happens postnatally as well. We discussed this in greater depth on Tuesday. Where does all this growth come from? How does the brain grow like this? Well, it's due to the action of nerve cells. So a nerve cell or a neuron is what you see pictured here. It's the main building block of the brain. At birth, the brain has a lot of these, about 100 billion neurons. And they communicate with one another. And that's what controls all aspects of our thinking, emotion, behavior. And there are three main parts of a neuron. We're going to use a model with our hand. This helped me learn it, so hopefully it helps you too. But what I want you to do is stick out your hand, stick it out straight in front of you, palm facing down, and we'll use that to go over the three parts of a neuron. So what your palm represents when you stretch your hand out is called the cell body. Let's see if I can use my mouse to click around. So it's here, if you can see my mouse. This part provides energy to the cell. This is important for the function of the cell, for it to stay alive. If you were to compare this to like your cell phone, this would be the battery. So it's keeping your cell phone going. In your hand model, if you look at your fingers, those would be what we think of as, or what are known as the dendrites. If you look at the screen, you can see that I'm pointing to these tree-like structures. They bring information into the neuron. So I said neurons communicate with each other. So information gets sent from one neuron to another, and it enters in the dendrites or our fingertips. So if you're thinking about a cell phone, the way cell phones used to look, where they had an antenna on them, this would be like the antenna. So it's bringing information into the cell phone. And these get bushier and spinier with experience. Your arm represents the axon. So every cell has an axon, and the purpose is to send messages out of the cell. So the dendrites sometimes are referred to as the listening end, and the axons referred to as the talking end. So it's like talking to other cells, essentially transmitting messages. So if you were to think of your cell phone, the axon would be the transmitter. And it's wrapped in this fatty protective coating that we call myelin. This develops as a child grows. If you think of like the protective rubber coating on the wires of your television, just like those wires, the myelin helps the messages move along the axon clearly. And as children grow, these messages, the myelin continues to mature, and it helps with communication of messages. And it can help the messages being sent more quickly. So I said that the neurons are the building blocks. So there are a lot of changes that happen in the neuron, both prenatally and extending into postnatal periods. So this is a visualization of the stages of the neuronal changes from around 1 week gestation to after birth. And the action of, or the changing of the neurons really takes place into early adulthood. We're going to briefly go over this again. We talked about this more in depth on Tuesday. But there's a particular sequence in which this takes place, and you can look. Neurulation, proliferation, differentiation, and migration are all stages that take place prenatally. So what those stages are, it's essentially that the neurons developed, and then there's a rapid growth in the amount of neurons. And then the neurons travel to what is their target destination, so where they're going to live. And so when they're traveling, some make it to their final destination and stay with us, and others die off. So they're not neurons that are going to continue on. So it's like kind of like taking away unnecessary things. And then once we get into the stage that's called synapse formation, these next stages continue on past the prenatal period and many into adulthood. So synapse formation, also known as synaptogenesis, what this stage is, it's the development of the point of contact between two neurons. So the first synapses are generally observed around the 23rd week of gestation, although peak production doesn't occur until like the first week of life. And there's typically an overproduction of these synapses, so we end up developing more than we need. And this is where experience really comes into play for the first time. So the other stages are typically driven by genetics, but when it comes to synapses, our experience and environment can shape the synaptic connections. So there's this massive overproduction prenatally, and then there's this stage called pruning, in which the unnecessary or unused connections are pruned, like you're pruning a bush, like taken away. So it's essentially your brain's way of organizing in an efficient manner. So all of this is highly dependent on experience, and serves as the basis of learning that occurs early in early years of life. And all of this highlights the importance of the protective and risk factors that we talked about on Tuesday. So early experiences are really important for development of the brain. Myelination is the final stage, and that's where there's that development of that fatty substance that surrounds the axon or the arm, if we're thinking about our arm-hand model. And myelin helps to facilitate neuronal activity and communication. And the timing of myelination is really dependent on the region of the brain in which it occurs. Regions of the brain for certain sensory and motor, so like more primitive functions that develop earlier are myelinated earlier in the process and completed around like the preschool period. But regions that are involved in higher order cognitive abilities like decision-making, planning, inhibition extends out until about adolescence or early adulthood. So I talked about this on Tuesday, but sometimes adolescents don't make the best decisions. Sometimes they make good decisions, but sometimes it's just like, why, what's going on? The brain is still developing during adolescence and adulthood, and a lot of those higher order cognitive functions like decision-making and planning and inhibition and organization are still taking place in adolescence and adulthood. So we also talked about some key components of brain development. If you would like more in-depth information about this, please visit the Tuesday webinar. But just briefly, brain development is sequential. We've already kind of touched on that. But essentially the lowest parts of the brain, or the most primitive functions of the brain, develop first. So the brain stem, which controls the basic regulatory functions like heart rate, blood pressure, and body temperature, develop first. It's mostly developed at birth. As you move up the brain, so seriously, like physically from the bottom to the top, the limbic system is one of the next areas to develop. And this part of the brain is really important for emotion and memory and motor function, attachment. And then if you move up the brain more, the cortex is what you think of when you think of what the brain looks like. That part has this concrete and abstract thought, and that's last to develop. Next, change in the brain is use dependent. So the brains have the ability not only to grow, but to change. And scientists use the term plastic since plastic is moldable. So essentially our brain is like plastic, it's moldable. Brain activity is activity dependent. So we've discussed this in the last slide. Experience really changes the foundation of the brain. Organization is based on experience. So we talked about how you're born with more connections than you need. And then what's interesting is between birth and like 6 years old, you develop even more connections. So we essentially go through these synaptic growth spurts. But these neural connections are reduced through experience. This helps make the brain more refined, and it produces a more efficient adult brain. And then there are critical windows of opportunity. So the brain organizes, it requires specific patterns of activity to occur at specific times of development. During these critical periods, the brain is extremely sensitive to input from the environment. And the critical windows are different for different functions and neural systems. So more basic functions like language, hearing, eyesight are very vulnerable during the first year, but more complex cognitive processes like decision-making and planning are later and extended into adolescence. Okay. So now that we have an understanding of brain development and the functions of various areas of the brain, which we talked about in depth on Tuesday, we're going to discuss how we can study this in humans. So we'll set the foundation for the methods of how we can study the brain, and then this will lead us to specific research on various conditions. Okay, so we're going to be talking about the field of neuropsychology. What is neuropsychology? So we've been talking about the brain. What does the brain do? This is really what neuropsychology is. It's the relationship between what's going on in the brain that produces particular behavior. The central focus of this field is examining human behavior based on the function of the brain. So in the field of neuropsychology, we attempt to explain the way in which activity of the brain is expressed in observable behavior. So what's going on in the brain that is leading to this hockey player being a good hockey player? What's happening in the brain when this child is studying? What mechanisms are responsible for human thinking, learning, and emotion? How do the mechanisms operate? And what are the effects of changes in brain states upon human behavior? So like, what's the effect of damage to the brain? There are two main areas of neuropsychology. There's research neuropsychology, and that's where I fall. Research neuropsychologists research the relationship between brain and human cognitive, emotional, and behavioral function, using both normal and clinical populations. So it's important to study, and I'm using air quotes, as normal. I don't really love the term normal. I don't know how normal is defined, but in typical development, typical children, typical adults, essentially to serve as a foundation for studying clinical populations. So that helps serve as a guide, as what we would expect the brain to look like and to function. And it helps us to study pathology if we have like a comparison. Clinical neuropsychology is the applied science of brain and behavior relationships. So clinical neuropsychologists have a degree in clinical psychology, and they work on assessment, diagnosis, treatment, and rehabilitation of patients across the lifespan who may have neurological, medical, developmental, or psychiatric conditions. So neuropsychology can help us to understand the impact of risk and protective factors, which we talked about on Tuesday on the brain. So this serves as a review, and then also as what neuropsychologists can study to help us to understand the brain. But essentially, the architecture of the brain depends on the influences of both genetics and environment. Genetics supply the basic plan for brain development. So essentially, these can help these provide the structure of the brain's architecture and supplies means for interconnecting nerve cells within and across circuits. The environment has a profound influence on shaping the capacity of the brain. This begins prenatally with the brain's needing an abundant supply of nutrients, and then any adverse prenatal environment may affect genetic plans, and this can actually happen postnatally too. And then experience comes into this as well. Experience refers to the interaction of a child with his or her environment. Healthy or stimulating experiences can result in brain architecture that's able to operate at the full genetic potential, but then possible negative experiences can influence the genes and produce adverse outcomes. All right, so how do we go about studying the brain? There are multiple different methods that we can do this. We're going to talk about two of the main methods. So the main ways that neuropsychologists study the brain are through neuropsychological assessment, neuroimaging, and then lesion studies. We're going to talk about neuropsychological assessment and neuroimaging. We're not going to go in depth into lesion studies. But essentially what lesion studies are is they provide a way to make comparisons of performance before and after like lesion or damage. So you're able to look at like the deficits that occur. We obviously don't go into humans and lesion the brain. This is done in an animal model. So if you like lesion one particular area of the brain, you can see like corresponding deficit. We also can study this in humans if there's like an accident or in the face of neurosurgery. So the most common ways that we study in neuropsychology, that we study the brain, are through neuropsychological assessment and neuroimaging. So neuropsychological assessment is the evaluation of cognitive function using a battery of standardized assessment measures. And these are some of the domains or the functions that neuropsychologists study. They include intelligence, academic functioning, executive functioning, which is that higher order thinking, learning and memory, speed of processing, motor skills, visuospatial skills, and language. So what typically happens, and this isn't across the board, but this is just a typical example. If you go into a neuropsychologist, this would be either pediatric or adult, is that the complaints would be presented from the individual or the individual and others. The neuropsychologist may perform collateral interviews, so asking like family and friends about functioning, review any available medical records, neuroimaging records, and then they could go one of two routes when it comes to testing cognitive functioning. They could go one route, what's called hypothesis-guided testing. So essentially what's suspected of going on in the brain, picking tests that are sensitive to that to get like a very extensive picture of the domains that are thought to be affected, or using standardized test batteries, which are, standardized test batteries are essentially a large set of tests that tap into a wide variety of skills and abilities. They were developed before the era of brain scanning. So giving this wide array of tests and seeing someone's function, that can help point to where in the brain may be affected. So you kind of just throw everything out there. They can be very, very long. And the individual will take tests in all of these different domains, and then you can see compared to controls. So compared to like a healthy population, if someone's falling below average, average, or above average in certain cognitive domains. These batteries have shown to be very valid, so and reliable, so consistent across time and measuring what they're supposed to measure. And they really give a very extensive workup of the individual, but they are very time consuming. And they lack some flexibility to assess like specific deficits. If you suspect like a executive function issue, executive function is the manifestation of the frontal lobe. If you suspect something like that, you may want to give tests that are tap a wide array of executive function. A battery wouldn't necessarily allow you to do that because it would just be kind of like a standardized set of all different domains, maybe not going in depth into a specific function. So we are going to do an activity. So what I want you to do is, if you are at your computer, you can open a Microsoft Word document, like a blank Microsoft Word document, or if you have a paper and pencil around you, grab it. I'll give you a little bit to get that set up. If you're listening to the recording, you can do this too and play along with us. So I'll give you like 30 seconds and then we'll get started on our activity. Alrighty, so no cheating here. I know you have your Word document open or your pen or pencil and paper, but no writing anything down yet until I say, okay? So what I'm gonna do, I'm gonna read you a list of 12 words. Don't want you to write anything down until I'm finished. When I'm finished, I'll say I'm finished and write down as many words as you can, okay? teacher, basketball, lettuce, dentist, tennis, bean, engineer, potato, professor, golf, corn, soccer. Okay, finished. Alrighty. So if you're using a computer, go ahead and delete what you just wrote. If you're using a piece of paper, either cover it up or flip that paper over. I'm going to read it one more time. When I'm finished, I want you to write down or type as many words as you can, okay? Teacher, basketball, lettuce, dentist, tennis, bean, engineer, potato, professor, golf, corn, soccer. Finished. Go ahead. Alrighty. So if you have it on the computer, delete it. If you have it on a piece of paper, just put that piece of paper to the side or trash it. We'll come back to this. But what we just did is kind of a neuropsychological test. It was part of it. I didn't do the whole thing for time's sake. But this is what a test for measuring learning may look like. Learning's very specific to the frontal lobe, so I'm essentially testing frontal lobe activity. So what we would do if we were really giving this test, well, there's two different versions. In one of them, we would give this 3 times, and so we'd read the list out loud, and this could be to a child or adult, and then ask the participant or patient to tell us as many words as they can remember, and then we do it again, and then ask them to tell us as many words as they can remember, then we do it again, ask them to tell us as many words as they can remember. And then we count the total words remembered, and then also look at learning. So from time 1 to time 2 to time 3, and then you could essentially take this information and compare it to norms, which are like standardized based on age, education, race, so essentially demographically matched individuals, and determine if individuals are scoring below average or above average on this immediate memory or learning. It's referred to as a learning or immediate memory task. Okay. Another way that we measure brain structure and function is through neuroimaging. So there's structural and functional neuroimaging. These both allow us to examine brain structure and function in vivo, so in living individuals. There's multiple different types of structural and functional neuroimaging. We're just going to talk about a couple examples, like the most widely used. CT, or computerized tomography scans, use a series of X-ray images and from various locations, and then they're combined to construct an image. These don't have the best resolution, but they're really helpful for visualizing the structural problem, like a tumor. Magnetic resonance imaging, or MRI, uses a magnetic field and produces an image that looks similar to what we see on the right. This has really good resolution. So essentially, the mechanism of the scanner itself leads to a production of energy from hydrogen atoms. And the magnetic field responds to this energy. It can tell what part of the brain it comes from. And then a computer can help reconstruct the image. And the image that I'm talking about is like what we're seeing here. This is used very commonly in neuropsychological studies. Functional neuroimaging allows us to be able to determine in vivo what parts of the brain are active at rest and during particular tasks. So PET scans are what produces those really pretty pictures that you see. It's what's on the left. Essentially, an individual is injected with a radioactive substance, and a very simplistic explanation is this essentially attaches to blood, and the blood flow is detected by the scanner. So when there's activity in the brain, that's where the blood goes. So the scanner is able to pick up what parts of the brain are active. And then a functional MRI or fMRI uses a magnetic field similar to a structural MRI. And it measures, the picture can help to determine where oxygenated blood is in the brain. So areas of the brain that are active, that's where the blood's gonna go to. So this allows us to have a picture of that. It's detecting the oxygenated blood. So it determines what areas of the brain are active, either at rest or during a particular task. This is sometimes, it depends what you're looking at, but sometimes preferred over PET because of safety and that there's no injection needed for this. All right, so those are two common research methodologies for studying the brain. I want to put this together. And hopefully, as we go into the next part, it'll explain why I'm telling you about this. But essentially, I wanted to discuss these methodologies as they're really important for our understanding of some pediatric diagnoses. So what I want to talk about from here is how we use these research methodologies to understand particular diagnoses, and then hopefully in understanding biological underpinnings and rationale for behavior, this can help increase understanding. So pediatric neuropsychologists, either researchers or clinical neuropsychologists, study a wide array of conditions. We're going to be talking about a few commonly studied conditions. By no means is this inclusive. I just wanted to give you some examples of neuroscience research in terms of some pediatric diagnoses. But brain injury, medical diseases, developmental problems are all studied. Some examples are toxic prenatal exposures, neurocognitive disorders, autism spectrum disorder, nonverbal learning disability, traumatic brain injury, motor disorders. And this would all, these would all be conditions that are examined by neuropsychology research. So like I said, we're going to talk about a few. Autism spectrum disorder, the diagnosis is based on the Diagnostic Statistics Manual, or the DSM. And what that does, this manual, it spells out defining features and symptoms of psychological disorders to help with diagnosis. Autism spectrum disorder is diagnosed based on deficits in two behavioral domains, social, communication, and interaction, and then restricted, repetitive patterns of behavior, interests, or activities. So when I say social and communication, essentially an individual may have difficulty forming social relationships, responding appropriately to social cues, sharing experiences with others, and participating in normal social interactions. Restricted behavior would be seeking sameness or repetition, obsessions with particular objects or interests, repeated phrases or stereotyped behaviors like rocking or clapping hands, or elaborate routines. This diagnosis includes previous categories of autism, Asperger's syndrome, and pervasive developmental disorder. Why do some individuals develop autism spectrum disorder? The exact causes are unknown. There's evidence for a genetic component. So on the slide, it says concordance rate of identical twins. So what I mean by that is the probability that a pair of individuals will both have a certain characteristic given that one has that characteristic. So in twin studies, if one has it, 76 to 88, depending on the study, it's been a range, will have that characteristic as well. There's also been studies showing that relatives have elevated levels of autistic traits. We talked about on Tuesday that genes and environment don't act alone. We don't think of it as genes versus environment. We think of it as genes and environment, so nature and nurture working together. So there's been studies showing that the environmental interaction with genetics, those factors may include perinatal complications, increased parental age, or exposure to infection, pesticides, and nutritional factors. And when I'm talking about this, in this case, and all of the cases, this does not apply to everybody with these various conditions. It's not across the board. This is just what research is pointing to as possible contributing factors. Diagnosis of autism spectrum disorder has been increasing. So in 2016, one child in 68 was diagnosed with autism spectrum. Prior to 1990, it was 3 to 6 children in 10,000. Not sure why. Could be due to an actual rise in incidence, or there's a theory that it's relaxation of diagnostic criteria, or most likely both. So what do we see in the brain of, I say many because this is not across the board, but individuals with autism spectrum disorder? What have the studies shown on average what we see? So there's some evidence to show that brain development is abnormally accelerated through early childhood, producing first brain enlargement, you could see by the image on the right, and then that's followed by a period of deceleration. So the clinical onset appears to be preceded by these 2 phases of brain growth abnormality. First, a reduced head size at birth, then a sudden and excess increase in head size between 1 and 2 months and 6 and 14 months. This abnormally accelerated growth may serve as an early warning sign for risk. Specifically, it's been shown that certain parts of the brain proliferate, so grow in the amount of neurons widely during early sensitive periods, but don't become interconnected with other brain regions, and pruned, so pruning off the connections in a typical way. However, this only fits some children. On Tuesday, we talked about the limbic system, which is important for emotion and memory, among other functions. There's been some studies showing differences in the size of the limbic system structures, specifically the hippocampus and amygdala, which we talked about on Tuesday. There's studies showing that some children with autism spectrum disorder have larger volumes of these structures than children without autism spectrum disorder. This may be reflect an increased activity of these structures and thereby altered emotional perception and regulation. The cerebellum, which point over here on the right, sometimes it's referred to as this mini brain. It's really important for cognition and motor coordination. It's been shown to be smaller in some individuals with autism spectrum disorder. And the degree of difference has been correlated with behavioral symptoms that have been shown in autism spectrum disorder. So this first condition that we discussed serves as an example of utilizing the methodologies that we discussed to understand the relationship between brain and behavior. So actually using neuroimaging to determine what we're seeing in the brain, and then using testing to determine how an individual is functioning, and then relating them to one another. All right, so attention deficit hyperactive disorder, ADHD, is characterized by inattentiveness and or impulsivity with hyperactivity. According to the DSM, which is that manual that helps us with diagnosis, symptoms must interfere with or reduce the quality of social, academic, or occupational functioning. Heritability is about 70%, so there's that genetic component there. But research has shown that it's likely that multiple genes are involved. So just because you inherit one characteristic does not mean that you're going to develop this condition, but it could be an interplay of multiple genetic characteristics in addition to environmental factors. Some environmental risk factors may include lead contamination, low birth weight, prenatal exposure to tobacco, alcohol, and other drugs. It's been shown that, well, the recent figure that I saw from 2016 is that 9% of children aged 2 to 17 have received an ADHD diagnosis. So what do we see on average, in the brains of individuals with ADHD. So you look at the, well, let's focus first on the frontal lobes, the prefrontal cortex. So there's been some evidence to show that the frontal lobes don't function as they do in typically developing children. This can lead to deficiencies in executive function. So the frontal lobe, which is here, I have my mouse on, is important for inhibiting behavior, organizing, emotional behavior, sustained attention. And there's some studies to show that there's this region of the brain is smaller, and the connections that go from this region, the frontal lobe, to other regions. And this has been shown to be associated with executive dysfunction, which are symptoms of ADHD. Research has also shown smaller volumes, similar to larger volumes are shown in autism spectrum disorder in the amygdala and hippocampus, and smaller volumes in ADHD. In the amygdala and hippocampus, and then also an area called the basal ganglia, which is important for cognition and motor function. It's located in the limbic system as well, so that emotional center of the brain. And also the brain as a whole. There's been other studies showing that there's reduced connection, there's disruption in connection between the right and left hemisphere, so the two sides of the brain. And that has been shown to predict symptoms. And research is really moving in the directionality of looking at the connections that run throughout the brain in the understanding of ADHD. Brain maturation, similar idea as autism spectrum disorder, but presents slightly different. But there's been studies showing that a number of children that are diagnosed with ADHD experience delayed brain maturation relative to their peers. Typically, we see this period of cortical thinning, so going through a stage of pruning. Around the peak levels around 7.5 years, but this isn't seen in children with ADHD until about 3 years later. So this finding may imply that some children with ADHD might eventually catch up to their peers, although others continue to experience symptoms throughout life. And then this next slide is essentially a visual image of what I was talking about. So there's been studies showing disruption in the frontal lobe, which is now it's on the right side of the screen. It's highlighted there, and that's been associated with some of the symptomology of ADHD. So both that particular area and then the connections that run through that area to other parts of the brain. And then also I had mentioned white matter disruption. This is probably taking us back a little bit to brain development, but there's two main components of the brain. There's gray matter, which we think of as like the main structures of the brain, like the hippocampus, the amygdala, the frontal lobe. And then there's white matter, which are the connections that go through different areas of the brain and link different areas. So if you think about it compared to a highway system, the gray matter would be like cities, and the white matter would be like the highways that connect the various cities. So if there's like a traffic jam, like in South Florida always, or an accident or anything like that, it's going to disrupt your ability to get from one city to another. That's similar to when we see white matter disruption. There's some sort of disruption that's impairing communication from one area to another. And that's what has been shown in ADHD, specifically the communication that takes place between the left and the right hemisphere. And this has been shown to be associated with behavioral symptoms. Major depressive disorder, unfortunately, this diagnosis does occur in children and adolescents. It's marked by periods of pervasive sadness that last at least 2 weeks, withdrawal from pleasurable activities, disturbances in appetite, energy levels, sleep, and cognition, and feelings of hopelessness, guilt, and worthlessness. Ever having been diagnosed with either anxiety or depression among children aged 6 to 17 increased from 5.4% in 2003 to 8% in 2007, and to 8.4% in 2011 and 2012. When we talk about major depressive disorder, we typically talk about adults. It presents slightly different in children. So in preschool children, compared to adults, we see similar behavioral and somatic symptoms like losing interest, eating poorly, but children don't often talk about their feelings of hopelessness or sadness like adults. As early as 3, some children express shame or guilt, claiming they are bad. They may act out scenes of death or suicide. The main things to look for are sadness and irritability. Children that are diagnosed with major depressive disorder are more prone to anxiety and anxiety conditions and more likely to develop major depressive disorder as an adult. We don't know the specific cause. There's, again, genetic and environment interaction. So there's been twin studies to show that the heritability is around 40%, but there's no specific gene that's involved. There's numerous that may increase risk for developing major depressive disorder, like the serotonin transporter we talked about on Tuesday, but there's no specific gene that if you have the specific gene, you're going to develop major depressive disorder. It interacts with the environment. Some environmental conditions that can increase risk are stress and prenatal nutrition. That's not an inclusive list, but there's some examples. So, similar to ADHD, there's been research to show lower volumes in both the hippocampus and amygdala, which again are limbic system structures important for emotion. Typically, smaller amygdala volumes present early in the course of pediatric depression and may predispose the development of this disorder, or perhaps more generally of childhood mood disorders. There's mixed evidence for hippocampal differences. Some studies have shown the hippocampus is smaller, others haven't. This is the joy of research. You have to do a lot. Each research study fills one piece of the pie, but we got to fill the whole pie to have an understanding. Hippocampus, we talked about on Tuesday. It's one of the areas that can be impacted by toxic stress. So the overactivation of the stress response system, which is why we may see differences in the hippocampus in major depressive disorder. And then also there's been research to show smaller areas or smaller volumes in the basal ganglia, which is also in the limbic system. And one specific study that's listed on the right showed that children that are treatment naive, so haven't received any treatment for major depressive disorder, have smaller volumes in this area, and it's related to the severity of depression. So the next condition is not as common, but it's severe. It can be very severe when it happens. There's been a lot of really interesting neuroscience research on it, and that's schizophrenia. It's diagnosed by at least 2 of the following to be present most of the time during a single 1-month period: delusions, hallucinations, disorganized speech, grossly disorganized or catatonic behavior, diminished emotional expression. Diagnosis is more common in male, and this typically is diagnosed in adolescence and early adulthood, and we'll talk about why that possibly is. So some theories for why that happens. I feel like I'm a broken record here by saying genes and environment interaction, but as I said, that we don't think of genes and environment as separate. We think of them as working together. So the susceptibility genes for schizophrenia overlap with some implicated in bipolar disorder and autism spectrum. And environmental factors can interact with genetic vulnerability. There's been higher rates in urban versus rural areas, lower socioeconomic status, marijuana use. We'll talk about the specifics of marijuana use, but and then prenatal environmental factors like perinatal complications, malnourishment, infection, or vitamin D deficiency. Again, this is not across the board. It's just what's shown in some individuals. Just quickly, if you look at the bottom part of this, on the left, you see risk of developing schizophrenia based on like relationship. And then on the bottom, on the bottom you see risk, and then you see the relationship on the the y-axis or the vertical axis. So a person's odds of being diagnosed with schizophrenia increase when closely related family members have the disorder. So the odds of being diagnosed with schizophrenia increase if you marry a person with schizophrenia. So that would increase the risk of your children developing schizophrenia, or in identical twins. Those are the highest risks. So what do we typically see in schizophrenia? It's associated with enlarged ventricles. So essentially it's this, we see the space here. It's where our nourishment for the brain is housed. And if you have enlargement in this area, you typically have a smaller brain. So it's associated with enlarged ventricles. On the left, you see a twin without schizophrenia. This is the top picture. On the right, you see a twin with schizophrenia. This picture shows the discrepancy between normal ventricles and the enlargement found in some people who have schizophrenia. Also, smaller hippocampal volume, so limbic system structure, hypofrontality, which is frontal lobe function. So the bottom picture on the left, you see a PET scan of a healthy twin. On the right, you see the twin with schizophrenia. On the right, you see less activity that's indicated by the red in the frontal lobe, which is located here, of the brain. So the frontal lobe, which is important for that higher order thinking, is less active in schizophrenia. And then also adolescent loss of gray matter. And then it's been shown that cannabis or marijuana use past first psychotic episode, once someone develops schizophrenia, produces even more loss of gray matter. So this loss of gray matter may explain why schizophrenia typically hits in adolescence or early adulthood. So teenagers typically experience a burst of cortical gray matter growth at puberty, followed by a wave of thinning. So again, it's like developing a lot of connections and thinning them off based on experience. And this extends into the early 20s. Compared to healthy peers, teenagers diagnosed with schizophrenia lose far more gray matter. It's sometimes described as a forest fire. So there's essentially this over pruning, we go back to neuronal development, or failure to produce sufficient synapses. So I know we're almost out of time, but I want to go back to our activity and then we'll conclude. So for the sake of time, we'll do this. So if you were writing on a Microsoft Word document, go ahead and delete what you had. Hopefully you already did, or grab a new piece of paper. And essentially, or what I want you to do is I want you to write or type as many words as you can remember from the activity that we had before this section, from that list of words. All right, so that's the other component of this test. So that particular part of the test is delayed memory. So it's after like 20, 25 minutes, and that's testing hippocampal, which we've talked about a lot, which is important for memory. It's testing hippocampal function. Don't worry about your performance on this, because we did not standardize this. You weren't supposed to be writing things down. There were other things going on, and just wanted to show you an example of the test. Alrighty. So what we've learned, advances in brain imaging have allowed us to safely examine brain development in vivo, so in living humans. These advances have helped us to further understand areas of the brain that are essential for complex cognitive abilities, emotion, and behavior. And that was shown in the discussion of the various conditions. Neuroscience research has really helped aid our process of neurodevelopmental processes and disease. And the attempt here was to increase understanding of biological underpinnings, to hopefully foster empathy and innovation in how we respond and educate children and adolescents. So I think it's helped me, and I don't know if this applies to everyone, but just understanding that there's specific differences in the brain in terms of the structure itself and the way that it's functioning that is associated with behavior. And understanding that, I think, for me at least, does help increase my empathy for individuals. And then understanding that can also help me to further my research in developing innovative techniques to help and respond to children.